So let us begin with morphology. Uh, in morphology, let us begin with the scale patterns, and all the morphological characters. Most of them are taxonomically important. And as I said earlier, that the snakes are not just a tube in a tube structure. There are so many variations in almost every single structure. I mean, single structure means say scale, say teeth, this kind of structure. You will find so many variations. Okay, and many of these variations are actually taxonomically very important. And as I said earlier, that as many characters, as many variations you can count in, or you can add in your matrix, using which you will be differentiating one snake from the other, it is better. Okay, so let us begin with scalation. And scalation begins with head, and head scalation is uh, most important when it comes to identifying snakes with scale patterns. I mean, using scale patterns. Uh, so say this is and these are different color codes that are being used to designate different scales. So this is the rostral scale, যেটা আমরা lip বলি, lips. This is the rostral scale. This is internal. These two are nasals, as you can find this nasal aperture there. So these two are. Let me use the laser pointer. So this is the nasal aperture. So this will be called obviously the nasal scale. One nasal scale, two nasal scale. So the scales in between two nasal scales will be obviously called internasals. So this is how we name scales. Now this is the frontal scale. So these two will be prefrontals. Okay, and as I said, this is rostral. That means our upper lip. Now sidewise, uh, again this is nasal. This blue one is called L'Oreal. Now, why this L'Oreal scale this particular L'Oreal scale is very important in identifying at least venomous and non-venomous scale to a great extent. Not I'm not saying that it is foolproof, but to a great extent, I mean, say almost all the deadly venomous snakes, almost all the deadly venomous snakes will not be having this L'Oreal scale. Okay, say or you may say the bona fide venomous families like Viperidae, like Elapidae, like Atractaspidae. These three are, and by saying Elapidae, I am actually including the sea snakes also. Okay, I mean, according to some classification schemes, you can find that they are using Hydrophidae as a separate family. But I mean, the classification that I follow there it is dealt with as subfamily under family Elapidae. So three bona fide venomous families do not have these L'Oreal scale. None of them. No snake in these families will be having this L'Oreal scale. So largely we can or loosely you can say you can actually identify non-venomous and venomous by the presence of this one. Presence or absence of this one. If it is absent, then it is likely to be venomous. But if it is present, it is not deadly. Ah, again, there is exception. There are certain colubrids which are having these L'Oreal, but still they are venomous. But those are exceptions. Okay. 90%, more than 90% cases, you will find that non-venomous snakes, or more than 95% cases, you will find non-venomous snakes will be having this L'Oreal scale, and the venomous ones will, I mean, they don't have this scale. Okay, now this is the eye. So this is the eye or ocular region. So these two will be preocular, and these three will be postocular. Okay, the light blue one preocular, these are postocular, and this is supraocular, and these three are infraocular. Supra, infra. And this is three means for this particular negative three, it may vary. The number and the shape of these scales may also vary, and that will add to the intricacy of the of your classification matrix. I mean, your taxonomic matrix. Okay. So this is how, and these are supralabials and these are infralabials. That means আমাদের upper lip এবং lower lip বলা যেতে পারে. So supralabials and infralabials, again the shape and the color and the scales that they are in touch with, all these things count while identifying a particular species or a particular family. Okay, so head scalation is very important. And this is the ventral area. This is called mental group. I mean, in later slides, you will find what is the mental group. I mean, the snakes that are most of the snakes actually do this. Most of the macrophagus snakes. Macrophagus means the snakes that will take on that will take in larger prey items. And for that, I mean, the snakes that will be dealing with large prey items will be having deep mental groups. Okay. We'll discuss later on when it comes. So and this is the typical head scalation of almost any sea snake, and as you can see, it is. I'm not saying this is patternless, but it is not as patterned as you can find here, as discretely patterned as you can see here. There are some irregularities like this. So this is a typical characteristic feature of a sea snake scalation, head scalation, plus the nasals, especially the sea snakes that will or that are pelagic. They will be having their nasal aperture on the dorsal surface or the nasal scale on the dorsal surface. Okay, and there won't be any internasals. If any snake head is lacking internasal and the nasal aperture is pointed upwards, that is a sea snake. Okay. And of course, the other scales are, you will find some amount of irregularity, some level of irregularity in the distribution of the scales, and most of the scales, other body scales will be of almost similar size and shape. Okay, regular size and shape. I mean, all the scales, be it dorsal, be it ventral, they will be of similar size and shape. So this is a typical characteristic feature. This is a typical characteristic feature of a sea snake. Now coming to variations again, as I say, there almost any structure that you can look at in a particular snake, you can find variations. If you have really that, I mean, if you are really interested to know what are the variations, you will find some variation. Like this, you can find this kind of a structure. If your observation is good, this kind of structure that is called scale sensilla. Not all the species are having them, but many of them are having, and it is believed that this kind of a structure help them in tracking prey, and they have something to do with that. And this is not fully established, but it is believed that they have something to do with the wave pattern, and following which they can. I mean, they are microwave pattern, not the microwave that we are using in our kitchens. Okay, the microwave waves that are being created by the movement of the prey item inside the. Say they have inserted this mouth inside a particular cave and crevices. That kind of a microwave pattern inside that crevices, that will help these snakes to identify or to locate each prey item inside the crevice. So it is believed. I'm not sure that this kind of an explanation is there, but this is, yes, one of the explanations, of course. Now, coming to the dorsal scale, how do we count dorsal scale? Now, in this particular figure, as you can see, that this snake diagram is slit open from the midpoint of the ventral scale. Actually, these two will make one single ventral scale. And the ventral scale is slit open at the middle. So half of the ventral scale is on the left side, and other half is on the right side. So we can count scales like this from one end of the dorsal surface to the other end of the dorsal surface that touches the ventral scale. Okay, so this is 17. Okay, in some other snake it may be 19, it may be 21, like that. Okay, and in most cases it is odd numbered. This will be either say 17 or 15. The last count will be say 15 or 17 or 19, 21, like that. Okay, and there are even number scale that I'll discuss later on. Okay, and again, this is another pattern following which you can actually count. This is another pattern. Anyway, this is one way, this is the second way of counting, and this is the third way of counting. Like this. In waves, you can count also. Okay. So this is how we actually do our practical job. Now, this is, coming to the scale texture. Now, see, at the graduation level, at least, I have seen some books at the local level. I'm not saying those are very authentic books at the international level, but these are local books, and there I have seen that in the taxonomic key they have used the texture of the scale to say whether it is a venomous snake or a non-venomous snake. So coming to the texture, at least you can find two types of texture. Either they will be ridged like this, or they will be smooth scales. Okay. Now they say that if the scale is smooth, it is likely to be a non-venomous snake, and if the scales are ridged, it is likely to be a venomous snake, which is not true. Absolutely not true. Now coming to the ridged scale. Now both these snakes are non-venomous. One is gopher snake, another is glossy snake. Okay, glossy doesn't mean that this is the glossy. They are talking about the glossy nature of the scale. Yes, of course, it is the glossy nature of the scale, but the name of the snake is also glossy snake. Okay. Now, both these snakes are non-venomous, but one is having ridge on their scale, and the other is not having them. Okay, so it has got, being killed or reached has got nothing to do with being venomous or not being venomous. Okay, this is another scale type. This is called button type scale. গুঁড়ো গুঁড়ো মতো মনে লেগে থাকবে যে গায়ের উপর গুঁড়ো 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 ডট 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 লেগে রয়েছে আর কি। So these are button pattern scale, and this is acrocordidae family. যখন taxonomy পড়াবো, you will know acrocordids, elephant trunk snails, snakes বলে এদেরকে। Now, coming to the ventral scales, this is a typical ventral scale, broad one. It is wider than longer. Okay, so this is a typical ventral scale of the snakes that I won't say that are practicing custodial mode of life, but the snakes that move fast, they will be having this kind of broad ventral scales. But coming to the, the scale that is, this is not a scale. This is the slux of skin. Now this is the ventral part of the skin. This is a typical python scale. That means the ventral scales are much shorter than, with respect to the size of the entire exuvia, the dimension of the ventral scale will be much less as compared to this one. Okay, so if you find this kind of exuvia in your, this kind of a slat of skin in any part of your garden or somewhere, this is a python scale. This is ventral scale. How we count 1, 2, 3, 4, 7. Now again, Coming to the anal plate. Anal plate means this beneath this plate you will find the anal opening, anal aperture. Okay. Now this is called the anal plate. Okay. Now this anal plate can be of two. I mean two. I mean they are of two types. Either they are entire. I mean there is no division, or they will be divided. So this is a single undivided scale. This is a divided pattern. Okay. So and again, there are books that write that some books say that if it is the entire one, I mean, if it is whole, then it is non-venomous or venomous, whatever it is. Because I don't try to remember what they say because that is bullshit. I mean, it has got no value. Absolutely, it has got nothing to do with being venomous or non-venomous. Again, it applies to this. Okay, and again, the subcaudals can be of two types: either they are divided or they are whole. Okay, again, it has got nothing to do with being venomous or non-venomous. Okay, but in books you will find this. So just ignore them. I'm not saying ignore them because if you ignore them, you will not get marks. But this is true ophiology. Okay, the things that are written there, those are wrong information. Absolutely wrong information. Now, they say anal subcaudal scale, and subcaudal means this is up to this is caudal and then subcaudal. Okay, so up to anal it is caudal, then subcaudal. Now, this subcaudal, say single row of subcaudal cells. Now look at the snake. This is rattlesnake, highly venomous, copperhead, cottonmouth. These are all venomous snakes. So this is venomous. This is also venomous. Okay. Now look at the scale. The anal plate is undivided. Okay, there is a series of subcaudals which are undivided, but there are subcaudals which are also divided. Same thing here. So in one snake you will get the proof that it has got nothing to do with being venomous or non-venomous. You can have subcaudals divided, you can have subcaudals undivided, you can be venomous, you can be non-venomous, it doesn't matter. And here you can find the anal plate is divided. Anal plate is divided, the subcaudals are also divided. Okay, and this kind of a scale pattern, subcaudal scale pattern, you will find in both colubrids and coral snakes. Now, in colubrids, you will find 90% of non-venomous snakes, but in coral snakes, 100% they are venomous. All of them are venomous, and many of them are deadly venomous. Coral snake. It comes under family Elapidae. Okay, we have got Asian coral snakes, we have got American coral snakes, doesn't matter. But the colubrids and the coral snakes both are having divided anal plate as well as double row of subcaudals. So again, it is another proof that being divided or not divided has got nothing to do, absolutely nothing to do with being venomous or non-venomous. Now, this is a true actual picture. Now, this is taken from a night snake and this is a western glossy snake. Both of them are non-venomous and these are entire, these are divided, this is divided, this is entire. So, this is a big proof that it has got nothing to do with being venomous or non-venomous. So, because they are both non-venomous, but they are having all kinds of variations in them. Now, coming to the identification, when I say I identify or we identify snakes, so these are the basic tools that we use to identify the snakes that we handle. This is a leg guard that keeps me alive when I deal with venomous snakes. This is and this is bite proof, or this is effectively bite proof, and these are sexing probes. Now, what are sexing probes? Sexing probe means the moment we catch a snake, the first thing that I do at least, I try to now I don't need to actually probe the snakes always to say whether it is a male snake or a female snake. But if there is any doubt, it can be cleared easily with the help of this tool, simple tool. These probes are inserted. I mean, in the next picture, you'll see how we do that. So then I'll explain. And these are some greasy material that we put on the tip of these probes so that it doesn't hurt while it is getting inserted inside the under the cloacal chamber. I mean the chamber that is under the cloaca to verify whether it is a penis or it is some uh, female structure. Okay. Now, now here, this is the tongue that is foldable here. These are all I've imported. These are all imported materials from America, and this is the tongue, foldable tongue. You see this is the head of the tongue. Tongue. These are press head so that it doesn't hurt while we catch the snake. Now, this is foldable, and this is the snake pin for pinning the snake because heading is very important while going on. I mean, studying the different scale structure and all these things. You cannot keep the snake head loose to let it bite you. Okay, so this is the pinning device with which we pin it. You can leave the snake also with the help of this structure. This end of the tongue. Now this, the sexing probe that use. This is the technique that by which we use the sexing probe. So this person is using the sexing probe. It is inserted. It is getting inserted. So this is the anal plate, and just underneath it, if it is a male, it will. The penis will be there deep inside, so the sac will be longer. And if it is a female, the sac will be smaller. It will end the moment you start inserting it. It will, it will inhibit the insertion. So you again, immediately you stop insertion because that is a confirmation that it is a female. This kind of sac they have. They also have sac, but that is of very small dimension. And this is the typical male leg যেটাকে বলা হয় আর কি, male caudal region having the penis. The inverted penis. So within the penis sac, it is getting inserted. Easily it will enter. Generally, by rule, the male caudal region will be the male tails. You may say male tail will be longer, much longer than the female. Now, coming to some freaky structures. As I said, there are variations. Any structure that you identify, you will find variations. If you observe critically, you will find variations. Okay, now these are freaks. This is not variation. This is very intriguing variation. Okay, now this is a Madagascan wine snake. Obviously, by name you can identify that it is found in Madagascar. Okay, Langaha madagascariensis. This is colubrids, but these are venomous. As I said, the colubrids, not all the colubrids are non-venomous. Okay, there are some colubrids at least who are venomous, and some of them can be deadly also. Okay, now let us come to this tentacled snake. The family is Homalopsidi. It is Arpedon tentaculatum. It is in Vietnam. Then you can find in Cambodia, in the rivers of Cambodia. This is a freshwater snake. It has got a nice story to tell. Later on, I'll tell it when it comes to the taxonomy of snake. Then I'll try to cover it. Tentacle snake. These are the tentacles. With the help of these tentacles, these are highly effective organs. These are highly effective organs, and these organs actually help them in catching their prey item easily. These are the two antennae with which they actually measure the distance of the prey item from their mouth. And in fraction of a second, with the help of these two structures, they will judge the distance and they will open their mouth and they will take in the prey. This is very fast. This is a very, very fast event. Okay, I could not share the video because it will take some time. And the video is available in YouTube. You can go through that. It's fantastic. I mean, they call it. I mean, eating the prey. The process with which they eat the prey. It is called C-start. It is called C-start mechanism that is being used by this Apheton tentaculatum or tentacle snake. So again, and these structures, these are not very. I mean, I don't know what is the utility of this structure. I have tried to decipher, but I didn't get the exact explanation or very acceptable explanation. There are some explanations some people have put forward, but I didn't find any. Maybe there's, I mean, with more research, we will find the exact uh, utility of this structure. Okay. Now, again, this is a gaboon viper. You can find the peculiarity in the scale structure. This is a fantastic specimen. This is a fantastic specimen. Means where this particular snake, this is a viper, of course, and these are found in Iranian desert. And look at the tip of their tail. This tail looks like a spider, and the as you can see the hue of their external coat, I mean the external body cover, it matches exactly with ambient structures. Okay, now uh, the way they move their tail, it moves like a spider, exactly like a spider, and just beside the movement area, they put their mouth, and these are bird eating snakes. And to eat these spiders, the birds will come and they will jump on the they will come directly inside the mouth. I mean, it is positioned in that way only. This is another intriguing structure function relation. This is turtle-headed sea snake. Again, modification of scale you can find here. This is sea snake, but this is non-venomous. As I said in the last class, that in almost every animal, in any animal group that you can possibly imagine or you can learn about, there are specimens, there are species that will try to act like a renegade, that will not follow the rules. People say set by nature, but I say set falsely by uh, our limited observations. Actually, there is no such kind of rule, maybe. So we come across these freaks, so-called freaks, but they are perfectly all right from nature's point of view. They are perfectly all right. They are not freaks. Okay, so they have these snakes have done away with their venom apparatus. Why? Because to avoid interspecific or maybe intraspecific competition for the prey item. I mean, food based competition. They have changed their trophic behavior uh, or trophic domain from being uh, fish eaters to being fish egg eaters. Now, when you specialize on eating eggs, you don't need teeth for that, because those, I mean, fish eggs are of very small size. So you do, I mean, for what purpose you will be needing those teeth, those fangs, having venom? I mean, to dislodge a prey, I mean, to knock down a prey, you need venom, you need fangs. But if your target is egg mass, then you do not need that extravagant structure, extravagant mechanism to, I mean, you don't need to have them actually. So they have done away with that structure. I mean, they have uh, very small fang size, if at all, and other teeth are also very small dimension, and their venom gland is almost atrophied, and whatever venom machinery that they are having still, this particular species, they have. I mean, if you go through their genome, they have uh, so many, what should I say, pseudogenes. They have so many pseudogenes in their venom genome profile. I mean, the part of the genome that deals with the venom production. I mean, different. They have, as you know, the venom is basically a cocktail of different toxins and peptides and all these things. Many other things are there. Venom biology আরো details এ পড়াবো. Now these venom, uh, they as they do not need an effective venom, so they are not actually in a mood to maintain the venom infrastructure. If you talk about their venom hardware means the fang, the venom gland, and the venom software also means the genes, the group of genes that will contribute their peptides and proteins into the venom. So all those genes, not all, many of those genes have actually turned into pseudogenes, which do not, I mean, these group of pseudogenes, they do not have any functional uh, product. So they do not contribute, so to say, inside the venom, whatever is left as venom. So they have a very low capacity venom or very not very effective venom. Now, there are some, I mean, though there are variations, there are some trends also. More or less, you can find they are maintaining this trend. Say boas, pythons, many vipers have irregularly arranged small head scales. This is 99% true for all the boas, pythons, and many vipers, of course. And cynopedians. Now, what is this cynopedian? Cynopedian means, I mean, to begin with, we can divide the snakes into the modern day snakes into two categories. One is Cenopedia, one is Cenopedia. Cenopedia is the more basal one, and the Cenopedia is the these we will call them modern snakes, modern lineage of snakes. Okay. Now in that modern lineage of snake, leaving the acrocordids, the snake with the button-like scales, those are acrocordids, like elephant trunk snake. So leaving these acrocordids, all the other cynopedian snakes or modern lineage snakes will be having symmetrically arranged head scales, or I mean, the structures are actually called plates or shields. Okay, the most snakes have longitudinal mental groove. The mental groove that I was talking then, the chin part, the mental groove. I mean, these will be found specifically in snakes that deal with larger prey items. So this kind of mental groove will be there in most of them, but there are a few groups which do not have this mental groove, like blind snakes, like dwarf pipe snakes, like goiters. Now, what is this goiters? Not all goiters. There are some goiters. Goiters means these are the snakes that prey on soft-bodied animals, any kind of soft-bodied animals. So soft-bodied, when a particular animal predates upon soft-bodied animals, they can be slug, they can be something else, any other soft-bodied animal, we call them goiters. Okay, now these goiters, these blind snakes, these dwarf pipe snakes, they do not have this mental groove because they do not deal with big prey items. Okay, so that is why they are not in a mood to maintain this mental groove structure. Okay. Now, some trends in scalation continue. Dorsal body scales typically overlap in diagonal, usually odd number rows, as I said earlier. In most of the snakes, we will find the dorsal scales in odd numbers, leaving these lousy species, and two or three more you can find if you search properly. In lousy species have actually even number count. That means it is not 17 like 19. It can be 18, 20, like that. Okay.
Okay.
Now
Now
range
range
from 10
from 10
to more
to
than
more than
90. When
90.
if you go to sea snake, you
You
can
can
actually
actually
have
have
scales,
scales,
dorsal
dorsal
scales,
scales,
up
up
to
to
100,
100,
say,
say,
90,
90,
100
100
dorsal
dorsal
scale
scale
rows.
rows.
Now
Now,
the
the
lower
lower
scale,
scale,
and so
so
these
these,
up
up
to
to
these
these
are
are
trends.
trends.
Okay.
Okay.
Now
Now
again
again,
there
there
are
are
certain
certain
tricks.
tricks.
The
The
lower
lower
scale
scale
rows
rows
of
of
males
males
of
of
short
short
sea
sea
snake,
snake,
Hydrophis
Hydrophis
cartus
cartus,
have
have
paired
paired
spiny
spiny
projections. I
projections.
have
I
a
have a
picture
picture
for
for
that.
that.
You'll
You'll
see.
see.
Have
Have
paired
paired
spiny
spiny
projections which
projections, which
probably help
probably
to
help to
secure
secure
a
a
couple
couple
whilst
whilst
mating.
mating.
This
This
is
is
one
one
explanation.
explanation.
There is
There is
another
another
explanation.
explanation.
Now, actually, this Hydrophis cartus group of sea snake, these are smaller size sea snakes, and they
They
are
are
found
found
in
in
the
the
what do you
what do you
call
call
মোহনা যেটাকে
মোহনা
বলে
যেটাকে বলে,
খাঁড়ি
খাঁড়ি,
কি যেন বলে
কি যেন
ওটাকে
বলে
the
ওটাকে, দ্য
estuarine
estuarine
region.
region.
Okay.
Okay.
Now
Now
estuarine
estuarine
region,
region,
there,
there,
I mean, to
to
avoid
avoid
the
the
mud
mud
from, I
from,
mean, these
structures,
structures, there is
there is
one
one
explanation
explanation
that
that
if they
if
are
they are
bogged
bogged
down
down
in
in
any
any
muddy
muddy
area,
area,
then with the help of these paired spiny projections in all the snakes, all in all the ventral scales, these structures actually help them to actually move a little bit faster in that muddy area. Okay, to give them a grip, give them a better grip so that they are not clogged down there. So this is another explanation, and pairing is of course another one. Now mid dorsal scales of the bizarre Javan Mars snake. Again, I have a picture for that. Javan Mars snake. It is this particular Javan Mars snake is also called the what do you call? Javan Mars
Javan Mars
snake.
snake.
Another
Another
name,
name,
common name
common
is
name is
there.
there.
It's not
It's not
coming
coming
right
right
now.
now.
So
So
these
these
Javan
Javan
Mars
Mars
snakes,
snake,
these
these
are
are
also
also
freaks.
freaks.
These are also having non-overlapping, as I say, the train is having what do you call the mm, I mean odd numbered scales, but this is and non-overlapping scales. But you can find one exception here, and another exception is these are non-overlapping scales, and that too of knobby nature. They are like knobs. They are more like knobs than scales. Okay.
Okay.
Oh
Oh
yes,
yes.
these are called dragon snakes. Dragon snakes. Okay, Javan
Javan
Mars
Mars
snake.
snake.
Now,
Now
most
most
snakes
snakes
have
have
enlarged
enlarged
transverse
transverse
ventral
ventral
scales,
scales,
but
but
blind
blind
snakes.
snakes.
So this
So
is
this
the
is the
trend.
trend.
Most
Most
snakes,
snakes,
90%
90%
of
of
the
the
snakes,
snakes,
or
or
95%
95%
of
of
the
the
snakes,
snakes,
you
you
will
will
find
find
they
they
are
are
having
having
transverse
transverse
ventral
ventral
scales,
scales,
big, broad ventral scales. But the blind snakes, most basal Althenopedians means those what do you call those Scolopedidia, Latinopedia. Latinopedia means the pythons and all these boas, all these things. Okay, so these most basal. I mean, the Latinopedia. There are certain Latinopedia which are even basal, more basal than the boas and pythons. Okay, they are talking about that group of Latinopedia. Some sea snakes have uniform size scales throughout the body. As I said earlier, there are in sea snakes, the dorsal scale and ventral scales are of almost similar size and shape. I've already said earlier. Now the ventral
Now the
scale
ventral
counts
scale counts range
fewer
from
than a
fewer than
hundred
100
optical slug
African
eaters.
slug eaters.
There's a
There's a
picture for
picture
this
for this
area.
area.
To more
To
than
more than
500 in
500
some
in
sea
some
snakes.
sea snakes.
Sea
Sea
snakes,
snakes,
I've
I've
given
given
it
it
in
in
red
red
because
because
99% of the sea
99% of
snakes
the sea
are
snakes are
venomous,
venomous,
and
and
90% of
90% of
them
them
are
are
actually
actually
deadly.
deadly.
So this
This
is
is a
a panoramic
panoramic
view.
view.
I
I
mean,
mean,
in an
in an
introductory
introductory
course,
course,
there are
there
many
are
more
many
things
more
to
things to
discuss,
discuss,
but I
but
have
I have
to
to
control my
my
intentions
intentions
and
and
my
my
desire
desire
to
to
bombard you with all those information because I cannot complete them. Then, so these are more or less the information for morphology. I mean, morphology. I mean, those areas of morphology that we use to identify snakes or to differentiate one snake from the other. Now let us look at the pictures that I mean the snakes that I have discussed already. The pictures. There are certain pictures. Now this is the Zachary species. That species, sorry, Zausi species. Zausi species means this one. That means the odd numbered rows except the Zausi species. These are the snakes that are having even numbered rows. So that is Zausis. Now this is the tiger snake. They are having 10 to 12 rows of scales, dorsal scales. So that is the tiger rat snake. Now coming to the short sea snake that I was talking about. So this is a muddy area. They're bogged here, bogged down here, and these are the projections on the ventral scales. Okay, maybe they're using them for from I mean for the purpose of getting out from this impasse. This is the Javan mud snake, the dragon snake that I was talking about. Now look at the scale. This is the enlarged version of the Javan mud snake. By the name 1095, these are found only in Java. Mm. So this is the typical dragon-looking snake, as you can see here. So these are freaks. These are freaks. From our point of view, these are freaks. And these are the variations. And always remember, today's freaks can be tomorrow's leaders. Okay, because Mother Nature is the most fickle-minded entity that you can ever imagine. Okay, the set of uh, priorities that are set to pass one species or to allow one species to live under the present condition with one mood change, the set of selection criteria will change, and today's best fit species can be tomorrow's extinct one. Okay, so these kind of freaks may then come to become the dominant species, the most successful ones. Okay, so the freaks are always there in any animal or plant world. Uh, as the nature is always changing, so sometimes today's freaks can be tomorrow's leaders. Now this is the African slug eater that I was talking about, the very small snake. African slug eater means is from the area, fewer than 100. African slug eater to more than 500 in sea snake. That means, I mean, this was discussed from the point of view of number of scales, ventral scales. Okay, so there are very few number of ventral scales. And this is an adult snake, as you can see. This is a very small snake, so the ventral scale number will also be less expectedly. So that's all for today. And for the question and answer session, you can ask me questions now, both from anatomy as well as morphology. Thank you.
Any question can be sent to the chat box also.
আর রাহুল একটু চ্যাট বক্সটা পড়ে দিস, আমি না দেখতে পারি না ইয়ের মধ্যে, মানে যদি কোনো কোশ্চেন আসে আর কি, প্লিজ লেট মি নো what are the questions. Is there any question? You can ask directly also. Bengali, in Bengali, English, Hindi, whatever you
তো চ্যাট বক্সে প্রশ্ন পাইনি।
Okay, if you want to ask any question, just switch on your mic and ask me whatever language you are comfortable in. Moitri, do you have any question? রাহুল মৈত্রীকে চিনিস তো? There is no question. You have understood everything that I have said. That is a very fine thing.
হ্যালো স্যার
হ্যাঁ হ্যাঁ
Yes, yes.
কোয়েশ্চেন
If you have any question, you can ask.
হ্যাঁ স্যার আমার একটা কোশ্চেন ছিল স্যার আপনি যে একটা জিনিস বলছিলেন যে আইডেন্টিফিকেশনের ক্ষেত্রে যে পোস্ট কোডাল সেলগুলো হয় মানে অ্যানাটোমির পর যে সেলগুলো থাকে
হ্যাঁ
ওটা ডিভাইডেড আর আনডিভাইডেড
হ্যাঁ
তো ওটা স্যার ভেনাম আর নন ভেনামাসের ক্ষেত্রে মোস্টলি স্যার এটা আমাদের ইন্ডিয়ান সাবকন্টিনেন্টের স্নেকের ক্ষেত্রে অ্যাপ্লিকেবল হবে আপনি যতগুলো বললেন
হ্যাঁ ইন্ডিয়ান সাবকন্টিনেন্টের ক্ষেত্রে অ্যাপ্লিকেবল হবে বিকজ this particular I mean this course is on snakes or ophiology. I've never said it is applicable for India only. The rules that I'm discussing here, it is applicable on a global scale. So for India, some rules may be applicable for India, but that is not a global trend.
হ্যাঁ
Okay.
ঠিক
So in India, you can find. I'm not talking about India only. I'm talking globally.
ওকে ওকে থ্যাংক
আচ্ছা চ্যাট বক্সে একটা কোয়েশ্চেন এসেছে আমি পড়ে দিচ্ছি হ্যাঁ সেটা একটু আনসার করতে হবে
বলো
Shaikh Hussein, he wants to know that recently vine snakes in India have been divided into many species. Is it based on morphology?
No, it is, yeah, of course it is based on morphology because every time it is, and if it is not, let me be clear, very clear about it. Let us not be concentrated or let our intentions not be concentrated only to publish papers. Okay, every time you do not have the chance of getting a snake, and every time you cannot go on identifying the snake with the help of their genes. Okay, so you must have, you should have. Even if you, even if these most of these vine snakes have been, I'm very aware of this incident. The vine snakes have been divided into so many categories now. But I doubt how many of them will be actually be there for a long time. Few more years. Maybe they will be clumped again in some group. Okay. Now, as a taxonomist, you should be looking for the differences in their morphology, not even in their anatomy. Because every time you cannot, the moment you get a snake, you go on dissecting the snake and look at its liver, look at its kidney, okay, then say, okay, this is the snake. Do not, that is a bad taxonomy. Okay, I'm not saying that molecular means these are touchstones, molecular tools and techniques that are being used to identify different snakes or different species of different categories, different domains of animals. These are very useful, but let us not be let us not come under them. Okay, let these tools be our tools. Okay, so there must be. You should always look for the morphological variations. Okay, then only it is a good taxonomy.
Okay, two questions have been found in the chat box. Although they are not the same, but I think they are very much relevant. So I am
Of course.
both of them. Moinak Mondol is asking that is there any particular reason for identifying the sex of a snake? And at the same time, Spirit Dash is also want to know that is it needful to detect sex of a snake before releasing in its habitat.
Come again, please. So is there any particular reason for identifying the sex of a snake? Of course, because as you have seen, the structure of the Madagascar snake, the freaky structure at the anterior end, there are different types. So if you go by the morphological character only, without identifying the snake sex, then you will probably come to the conclusion that these two are two different species of snake. So you always there is not always there is in many species of snakes you will find sexual dimorphism, and this is of one pinnacle. The snake that I discussed here, the Madagascan species, Langaha madagascariensis. So they are in their pinnacle. In terms of having a tricky structure, sex-based differences. So that is important. It is very important to identify a sex first, then go for the other structures.
আচ্ছা, অভিরূপ দাস জানতে চেয়েছে, is it needful to detect sex of a snake before releasing it in its habitat?
No, for releasing purpose, you don't need to detect the sex. Well, what for? I mean, why you should go for sexing for the purpose of releasing only? It is of taxonomic importance. It is of taxonomic importance.
আচ্ছা, অরিজিৎ দত্ত is asking that how impactful is morphology for the identification of polymorphic snakes?
Of course, for polymorphic snakes only, identification on the morphological point of view is important. Polymorphic snakes, of these polymorphisms. So this is the panorama of morphs that you study by studying morphology. Okay, if a snake is polymorphic, that does not mean that you are neglecting morphology as these are the single genome is giving you different types of morphs. So why should I concentrate on morphs? So while considering its morphology, consider all the morphs. Consider all the morphs. Say this morph is found in Barakpur region. The other morph is found in Odisha only. It is important, always important. Polymorphism is important, and these polymorphic doubts, the snakes that are polymorphic, that means polymorphic means they are having the same genome, but they are having different morphs in different regions of the country. It happens. And for that matter, you need those touchstones like different molecular tools. But do not, ওদের গোলাম হয়ে যেও না। What I am trying to say is ওই টেকনিক্স গুলো গোলাম হয়ে যেও না। Morphology and anatomy, these two are still very important tools, you may say, tools to identify majority of snakes, majority of animals till then.
Okay. Aditi Kamilla is asking how scale sensilla help in tracking prey. Is there any
No. Till date, I do not have any explanation. I've just mentioned what I've learned. I've just mentioned it. I do not have the explanation because I haven't come across that explanation. Okay. So that is a structure that they say they might help them in tracking prey in a closed environment. Those microwaves that will, with the help of those structures, they will be measuring or they will be gaining those microwaves generated by the movement of the prey item in that small area, and maybe it will help them in locating the prey in that very small domain. This is one of the explanations only.
Any other question? The speaker can directly interact, the participant can directly interact with the speaker.
Any language you are comfortable in, Hindi of course, it should be Hindi. Those participants, those who do not understand Bengali, you may speak in English, you may ask questions in English or Hindi, whatever it is. Okay, I'm not very good in Hindi, but I'll try.
Hello.
Yeah.
Hello.
কে বলছে?
এই আমি পিঙ্কুদা বলছি সৈকত।
হ্যাঁ বলো।
তুমি তো জানো আমার কোন রকম জুলজিক্যাল ব্যাকগ্রাউন্ড নেই।
আরে কিছু ম্যাটার করে না, বলো না।
আচ্ছা, আমার প্রশ্নটা কিন্তু খুবই মানে একদম প্রিলিমিনারি, মানে হয়তো এই ফোরামে করার মতোই নয়।
আরে তুমি বলো না,
সবাই জানে, আমি জানি না।
তুমি বলো না, বলো না।
ব্যাপারটা ব্যাপারটা হচ্ছে যে সাপ তো কোল্ড ব্লাডেড।
হ্যাঁ, হ্যাঁ,
তো ওয়ার্ম ব্লাডেড, আমরা ওয়ার্ম ব্লাডেড আর সাপ কোল্ড ব্লাডেড।
হ্যাঁ।
তো সাপের কি এই কোল্ড ব্লাডেড হওয়ার জন্য সাপের কি কোনো রকম অ্যাডভান্টেজ আছে?
না, না, অ্যাকচুয়ালি এটা খুব সাবজেক্টিভ হতো না, যে কোনো কোন আমি আমি ফার্স্ট ফার্স্ট এবার তোমার ইয়ে মাইকটা বন্ধ করে দাও।
হ্যাঁ, হ্যাঁ।
Okay, first, let me translate in English. What people ask me that I mean, these snakes are cold-blooded. So, is there any advantage in being cold-blooded? Of course, there is advantage. Now, these are, I mean, if you there are advantages in being cold-blooded, and there are advantages in being hot-blooded or warm-blooded. I mean, it is the area. It is the region. It is not applicable for snake only. It is applicable for any cold-blooded animal. There are lizards, there are turtles, there are insects. There's a whole bunch of cold-blooded animals. Now, these cold-blooded animals in regions that are not, I mean, the warm-blooded animals will spend huge energy in maintaining being hot-blooded. So those areas will not be very welcoming for the hot-blooded animals, and they will have to maintain so much of energy and so much of anatomical structures, morphological structures to maintain the temperature. That is the topic of thermoregulation. Okay, and if you can thermoconform to some extent, so you can be happy in many climates. Of course, you will have your limitations. You will have your movement limitations. You will have to restrict your movements in the comfortable temperature zone. Okay, the hot-blooded animals do not need that. But the hot-blooded animals, I mean, with those huge structures for maintaining the hot-bloodedness, they will not be in a position to venture into those caves and crevices or small areas where these cold-blooded animals can enjoy. Okay. So that is the advantage of cold-blooded sometimes.
হ্যাঁ ঠিক আছে ঠিক আছে ঠিক আছে। আমি পরবর্তী ক্ষেত্রে মানে এই কোর্স শেষ হয়ে গেলেও হয়তো পরবর্তী ক্ষেত্রে
লাইফটাইম এনিটাইম।
আর তোমার তো বাড়ি ব্যারাকপুরেই, আমিও ব্যারাকপুরেই থাকি।
ও আচ্ছা আচ্ছা, ও এটা জানতাম না। থ্যাংক ইউ।
আমি ব্যারাকপুরেই থাকি, মুক্তপুকুরে থাকি, মুক্তপুকুরে।
Any more questions, anyone?
No, we are not find any question in the chat box. I think there is no question from the participant end.
Okay. Then we can
end the session.
Okay.
Thank
you everyone.
Tomorrow we shall meet in the same
স্যার একটা
time.
প্রশ্ন আছে
বলো।
স্যার, আমি হচ্ছে ভূতত্ত্ব বিজ্ঞানের ছাত্র, তো স্যার আমার এই সাপ নিয়ে একটু ইন্টারেস্ট আছে, কিন্তু স্যার আমার মানে সমস্যা হচ্ছে কি, আমি এগুলো আইডেন্টিফাই করবো কিভাবে, মানে স্যার সাপগুলো চিনবো কিভাবে?
সাপগুলো চেনার জন্য এখনো তো আমরা সেই ডিসকাশনে ঢুকিনি। আমরা কাল থেকে শুরু করবো সেটা, মানে ট্যাক্সোনমি যেটা, or maybe you can say diversity, the world, the actual world of snake, how many types of snakes are there. এটা এখনো শুরু করিনি আমরা, we have not yet started.
আচ্ছা জি স্যার, তাহলে আগামী ক্লাসে শুরু হবে না তাই?
একদম মানে কাল শুরু হবে।
আচ্ছা জি স্যার, ধন্যবাদ স্যার।
ঠিক আছে, তাহলে আজকে মনে হচ্ছে এই পর্যন্তই নাকি? Any more questions? No. ঠিক আছে, তাহলে শেষ কর নাকি?
হ্যাঁ আজকে তাহলে শেষ করে দিচ্ছি বুঝলি, কালকে কি সময় আমাদের আবার সেশন শুরু হবে। সো উই আর এন্ডিং দ্য সেশন, closing the session, and tomorrow we shall meet in the same time. Thank you Mito, thank you Shoikat, thank you everybody, all the participants. আমি তো বন্ধ করে দিতে পারেন স্ট্রিমিংটা।